So I will be talking about my Zika virus research so far what I did since 2016, and I'll be mainly focusing on how we what the questions we asked and this how we designed the experiments. And in particular, this virus is the same virus which it spreads from the same mosquitoes which spread the dengue and chikungunya. So it always remains a problem to the world as well as the country as well. And so last year we didn't have any problem with the virus in the country, but we never know we need to be prepared and we need to do extensive research on this. So that was the motivation why I started and I am continuing this research on it. We asked very big questions on this and basic questions, how to think about this. So our first question was how a very limited pool of viral proteins are able to do very well established human defense systems. Because this virus has only 10 proteins, and with only 10 proteins, it is able to hijack the very well established human defense systems of the protein we can say, which is more than 20,000 proteins in humans. So, of course, there should be some collaboration among the proteins of the virus. So, the next question is how they are collaborating. Next question was what is the disorder function paradigm? Because we focused on the protein conformation. And on protein conformation, there are multiple conformations like something is intensely disordered, something is ordered, something aggregates. So what are different combination of the structures on there? What is the drug discovery possibilities on this on the viral targets, and how they collaborate for the pathogenesis and survival, how the replication complex is formed, and also about the virus. So I will just go about the snapshots and not go very detailed. So initially we cloned and purified and tried to purify many proteins of this virus, and we are successfully purified immunoprotein, helicase, protease, and NTLs, and also we are in between this NS1. So the technical aspect was three conformations, like what is the disordered conformation, what are the ordered conformation, what is the annulled conformation, and the drug discovery. So these are the two aspects we typically handle in this process. So the first thing was talking about the intensely disordered proteins. So we are very well aware about the structure function paradigm, which is very well established since 60 or 70 years. Because if something is structured, then they are functional, and this is called structure function paradigm, very well. But recently, since 2 decades, there was discussions about the disorder, that there are proteins which will be intensely disordered in nature, and still they are functional. And there was a dogma like if something is, there are so many proteins in the protein which are disordered and they still functional, then of course there is a shift in paradigm, and this is called disorder function paradigm. And generally, disorderness helps out in increasing the capture radius, like the concept raised by which understood the fly casting model. So if something is disordered, then they have more capture radius and they can do more protein interactions. On Zika virus, we investigated first the moment we started this research, and we started with the computational study, and we predicted the intensity disorder. And we found that on the protein junctions, there are disordered regions, or the linkage sequence, or the trans and loop sequence, and these regions were very important for the virus maturation. Further, we tried to experimentally see some of the predicted things. We found that in the capsid, the N-terminal region was to be disordered, and then when they synthesize the peptides, we found that this is intensely disordered, which is giving the peak negative recipe receiving spectrum at 130 to 100 range, which is the characteristic peak of a disordered protein. But again, the question was, if what about the disorder function paradigm? If it is something with disorder, if it is lying useless, or it is having some use, or it has some implications. So we thought that, okay, what this capsid does? Capsid actually does liquid binding, RNA binding, and ion encapsulation, packaging, and all this. So we thought, okay, let's see the conformational change within lipids. And we found that with the DOPS, it formed the alcohol. We synthesized the peptide of an S4A and terminus region, and we found that, yes, there is also conformational change upon interaction with the lipids. And these were supposed to be to work a good way, because these non-structural proteins, they the non-structural proteins are responsible for the replication complex formation, and they interact with the lipids, and then they uh, uh, do the create the environment for the replication changes. And on increasing salt proteins, we produce the couple of proteins and DNBs and glucose, and all this. So now let's go for the second conformation, which is the ordered conformation or the native conformation, you can say. So for that, we have taken the example of Zika virus helicase enzyme. We purified this enzyme and we performed this enzymatic assay. We found that the enzyme is active. We performed both the ATPase activity and the helicase activity using the molecular beacons, and we performed the pH dependent, whether means what is the suitability, what is the conformation of this enzyme. We performed the equilibrium experiments and the stability experiments. And we found that the viral protein stability is much lower as compared to the very well established global protein systems, because the global proteins are in the range of 20 to 60 kJ per mole, while this helicase enzyme is 7.9 kJ per mole. So we found that yes, these viral enzymes are not very stable, and that makes sense because they work in collaboration among each other. Because once the whole replication complex machinery builds up, and then only they function. So and further we perform the folding function relationships, and we also performed the thermodynamic experiments to understand the thermodynamics of the system. And we found that the TM of the helicase is very close to the physiological body temperature. And there was a question in our mind that how this more highest giving high, and the highest activity was also closer to the body temperature. So to understand that why the highest activity is coming at the near the melting temperature, we performed the morphodynamic simulations, and we found that the ATPase activity is given by the P loop, and the P loop gets more open up upon that increasing the temperature, and that is the role of these small loops or the active sites of the ATPase. So this is one of the aspects we did. Then we focused on the third conformation, which is the aggregation, the protein aggregation among the virus. And for that, we have taken the example of AD3 protein. We found that capsid anchor and ganglion proteins. So these are the three systems which aggregates and which has never been reported in any clinics. So these are the new findings what we found. And we verified the AD3 domain, and we performed the basic characterization of the cell structure. And we found that okay, this AD3 protein is aggregates, and this was this is a very different finding in the cellular system. So this is the third confirmation we focused, and then we are focusing on the drug discovery aspect against these products. So this is the fourth aspect of study we did on this on the proteins. And for this, we have verified the Zika virus NS2 protease complex. And we performed the enzyme assay inhibitions, and we performed actually the high throughput drug discovery using the Sodium software. And in the top 5 compounds, we found that hydroxychloroquine is there, and apart from that, there was more compounds. And for Zika virus, this is important that this is a major problem for the pregnant women because it causes microcephaly in the newborn babies. For the adults and for other people, it causes fever, which can be controlled easily. But for newborn babies, it is often lethal, and that is the reason it is important that the compound or the drug should be safer to the pregnant woman. And for that, we have taken the hydroxychloroquine, we performed the docking simulations, we performed the reduction, and then we performed the enzyme inhibition assays on Zika virus protease, purified enzyme. We found that the K value was 92 micromolar. And then at this stage, we communicated with Professor Microcephaly from Washington samples, and we asked that can we do the antiviral assays on the virus in your lab. So she performed the antiviral assays, and then we completed the full story, and we found that okay, hydroxychloroquine integrates the Zika virus protease enzyme, and this is how we leave the story. So that was one of the drug discovery snapshot. And apart from this, we also discovered that EGCG, the green molecule, inhibits the Zika virus helicase enzyme. Apart from this, we also worked on the envelope protein of Zika virus, and we found that there is one compound from a library which inhibits the envelope protein activity. No, an envelope protein folding back mechanism. And for that, one of my student or the PhD student went to University of Leeds, where he performed the antiviral and as a leading developer, and we could finish those experiments, and we have communicated that also for a publication. So these are a couple of publications on the drug discovery aspect, and I'm thankful to so who helped in antiviral assays and some theoretical studies were done by mosquito and study, and also the intelligent partners. I thanks to my students who are very talented students. Uh, Nitin is thankful for the meeting with which he could perform the integral essays in University of Leeds. And essays as a Fulbright fellow. Fulbright the final of his thesis will complete in USA and performing the integral essays. And media and media colleagues to if you ever work, and these are the people who are the real responsible people who produce the data and we could lead to the systems. Thank you.